My name is Matt. I'm the COO and co-founder of Kanvas Biosciences. First of all, I'd like to thank everyone here for taking the time to chat. And secondly, I'd like to disclose that I'm not a PhD scientist. I'm actually an MD and medical microbiologist by training. And frankly, despite this terrible pandemic, I was actually having a good time. I enjoyed my career, and I wasn't looking for a change. That is, until my buddies, Hao and Ewan at Cornell, came and spoke to me. They told me that they found a brand new way to detect and identify bacteria, a way that would have lasting implications in the way we understood and leveraged the microbiome. So, for those of you who may be less familiar, the microbiome is the collection of bacteria that you have from your gums to your bum. And we've known in the scientific community that the microbiome is really important. But it's actually started to gain more and more importance in the lay press as well, and for good reason. For every cell in your body, there are actually 10 bacteria. And if the last 10 or 20 years were focused on understanding the human genome, the next 10 or 20 are certainly going to be focused on understanding and utilizing the microbiome. And it's because it's a critical part of human health. It's linked to things inside your gastrointestinal tract, things like colon cancer and inflammatory bowel disease. But it's also linked to things outside of it, like diabetes, high blood pressure, high cholesterol, even things a little bit further removed, like Alzheimer's and human longevity. And the problem is that we really haven't been able to properly understand it. We haven't been able to leverage it, because the way we currently identify bacteria is limited. If you think of either culture or molecular-based assays, they're either too slow, they're too expensive, and most importantly, they do not give you any insight into the spatial arrangement of microorganisms. And what I mean by that is where bacteria are located relative to one another, and most importantly, where they're located relative to the human host. And the microbiome is just like real estate. It's all about location, location, location. And so in response to this, we developed a transformative platform that we licensed out of CTL that would have lasting implications in the way we understand the microbiome, the way we do microbiome research, and the way we develop microbiome treatments. Our proprietary solution is something called Hyperfish, which allows us to identify, to quantify, and to spatially position bacteria with single-cell resolution at a fraction of the cost of competing technologies. So the way it works, in very lay terms, is that we take advantage of known bacterial sequences. These are already readily available. We then apply specific tags, which is our way of painting the bacteria, which is why our name is Kanvas. We then apply the specimen to a microscope. That part literally takes 1 minute. And then we have a machine learning algorithm to automatically interpret the data for us. So there's no technician looking at cells, counting cells, judging colors. And from the moment we receive a specimen, we can give you a definitive answer in under 2 hours. So just to put things into perspective, the way it works is we first start by taking an image. We then give you an ultra-precise cell count. And from there, we're able to taxonomically tell you who's in your specimen. So Hyperfish has several sustainable differential advantages. The first of which is that we're an order of magnitude cheaper than competing technologies. Sequencing runs are $200 to $300 a pop, but we can run a single specimen for $24 at scale. Now, with some further engineering tweaks, we can bring it down below 20. We have high throughput capabilities because we only require 2 hours to give you a result. We can give you single cell imaging, and we can identify over 1,000 species per image. And because the human body only has 200 to 300 different species, we're comfortably above where we need to be. But the biggest differentiating factor is that we can provide an access of information that no one else can. We can tell you where the bacteria are spatially positioned. And that's the reason we're going to leverage our technology to really transform microbiome research and associated drug discovery. Currently, we operate as a service-based model, and we're doing some early work with companies like Merck and Takeda, and this is why. So this is a photo of the colon, and on the top right, those gray cells, they represent host epithelial cells. And Merck and Takeda, they're developing next-generation colon cancer treatments, inflammatory bowel disease treatments. And they need to know which part of the microbiome is interacting with the host. But if you looked at other technologies, they could tell you maybe who's in the stool, in what quantity. But we're the only ones who can tell you who's important. And so that's why we've got early pilots and signed contracts with large pharma, biotechs, academic labs, because these partners need access to our technology to be able to really move their drug discovery platforms forward. We were fortunate enough to raise a $1 million pre-seed round at the beginning of the year. Over the last couple of days, we just wrapped up a wild seed round. That was a heck of a process. And now we're looking to really transform Hyperfish into actionable tools to really help move microbiome research and drug discovery forward. So my name is Matt. I'm the co-founder of Kanvas, and I'm happy to take your questions.
Hi. So I have a test kit that's been on my shelf for 2 years from a company called Viome, which I've clearly never used before. It's still there. So this technology that you're speaking to seems to go more only in a clinical field space aside to the consumer, or can you speak a little bit more to the path of your technology to either consumer or who your real consumer is?
Right. So first of all, I think Viome's a great company. But if you saw in Crunchbase a couple of days ago, they did a major pivot because they realized that direct-to-consumer wasn't working at all for a variety of reasons. Chief among them is the fact that they give you a result, but it's frankly uninterpretable. And so I'm glad you didn't use your Viome kit. Where we are today is the opportunity not to necessarily go to consumers because the microbiome science is really evolving. It's in infancy, and we really are a science-first company. And so who we're targeting right now are academic labs, biotechs, large pharma, people who are doing actionable research to really help determine how do we use those results in the microbiome. Moreover, we're adding way more information than a Viome test. A Viome test is a sequencing-based assay. It tells you who's there and some degree of quantification. We can say who's there, who's alive, and more importantly, where they're located. It's a fundamentally different thing.
Any other judges' questions?
I have another one. There also seems to, from the Viome lens or microbiome as a lay person, there seems to be some controversy about the relevance of microbiome in the medical community. Can you speak on that?
Yeah, I think that there is controversy because previously when you took a stool specimen, you had an entire collection of bacteria and the data are frankly uninterpretable. We know that only a fraction of the microbiome matters, the fraction that interacts with the human host. And prior to Hyperfish and prior to Kanvas, that information was unknowable. We're literally carving the market out now because we're the only people capable of offering that type of information.
That was excellent, by the way. So I'm curious on the investment pitch. It sounds like you have one-of-a-kind technology here, but you also have lower costs. So how are you kind of pitching that to investors?
Successfully. And so we've received a term sheet from Triple Impact Capital. DCVC, one of the larger VC firms, came in, added some sizable check to the round. It was widely oversubscribed, so something's working.
I meant more from the perspective of, are you focused on the cost side or one-of-a-kind technology, or kind of what's the leading driver?
Yeah, honestly, I think it's a bit of both. So right now, the way we're pricing our specimens, we're charging people over $1,000 a pop because no one can do what we do, but it costs us $24. So the margins are pretty substantial. And so to the financially minded investor, that's an immediate win. And then to the deep tech or the biotech investor, they recognize that we have the opportunity to do something special, something that's never been done before. And we really have the perfect platform to expand on this. And the sky's the limit because no one really understands the microbiome until we've been able to really give them the tools to do it. So both sides of the investing coin is really well fulfilled.
And you mentioned the colon is where you currently have agreements with Takeda and Merck, right? So, like, what other where do you see the growth, other areas where this technology is going to come into play?
Yeah, so we actually have a wide range of applications from obviously microbiome research to agricultural applications. Actually, the very first specimen we did was, to be a fun piece of trivia in 5 years, but it's actually a piece of lettuce because there's a bunch of agricultural companies that really need to understand the microbiome of plants, and that includes lettuce, alfalfa sprouts, a few other things like that. So they send us all these specimens to really try to figure it out. But the other major thing, and I just didn't have time to do this in a 6-minute pitch, is really our applications in clinical diagnostics. Because Hyperfish is so fast and so proficient at identifying bacteria, we think we have a really unique shot at helping patients in the hospital. And we've actually started some preliminary work with folks at Emory University to use Hyperfish there. So I'd be happy to expand on that at another point, but that's another major market that we think we can really help address.
That's awesome. Any other questions from the judges? Audience at all? Time for one or two? Oh, we got a couple over here on it. Thanks.
Hi. So I actually just recently learned about microbiota in my biology class, and I know that there are individual differences in terms of like your genetics and your environmental factors. So how does your technology account for those individual differences? Yeah,
that's a really great question. There's no doubt that it's a really complex interplay between the environment, the host, and the bacteria. And so the way Hyperfish works is that we're really proficient at tagging bacteria, but there's no reason that we can't tag host cells, and we do that as well. So if you had another company that was interested in situ hybridization, they are focused on, say, mammalian cells, but it's very, very difficult to translate to bacteria because they're smaller, the genes are more compact, they're compressed, etc. But the reverse isn't true. So our technology works really quite well in mammalian cells. And so we actually have a unique opportunity to understand the host and the microbiome in concert, specifically because it is such a complex area.
And time for one more, right in the back, right behind you.
So is the business model also incorporate recurrent revenue? Because as you initially identify, quantify, and map the microbiome for therapeutic treatment, that's also going to have adverse effects. Is that something you're going to be doing sequential study of this, and you're taking that into account, I imagine?
That's exactly right. So one example is a company called Federation Bio, and they actually approached us because they're developing a microbiome as treatment, meaning you would ingest a combination of bacteria to treat high blood pressure, high blood sugar, etc. And the FDA mandated to them that they verify in their microbiome who's alive prior to human administration, right? They want to know who survived your manufacturing process. And there's no current technology out there that can give you a rapid answer in a matter of a few hours. Culture-based assays take 4 or 5 days for anaerobes, and sequencing-based assays, molecular assays, can't tell you who's alive, right? And so we developed Hyperfish specifically to address that. And that's one of the reasons we can get recurring revenue through these contracts with these companies that require a fixed amount of testing, either for a clinical trial or for a specific research project.
Perfect. Thank you so much. That was great.